Our next speaker is Dr. George Sutphin. George, I can pull this up while you start.
Hey everyone, I'm George Sutphin. I'm an associate professor, molecular and cellular biology in the Bio5 Institute at the University of Arizona. I'm down in Tucson at the Bio5 campus. For disclosure, I'm also a co-founder of a company based around some of the technology we're going to talk about here, Sutphin Biosystems. Sure. Yes. Don't like to chase us, I guess. There we go. So I think a lot of Noah's introduction really also sets up some of what I'm going to talk about, because we're really taking advantage of these a lot of these same underlying ideas, this universality of aging, and how can we leverage that to make new discoveries in aging science that we can maybe turn into therapeutics. And I'm going to be specifically talking about using C. elegans. So C. elegans are a roundworm, not life-size. This is a C. elegans, but they're about 1 millimeter long. This is one of the advantages. They're transparent, and in the image you can see several proteins labeled with fluorophores, which lets us get molecular data about these worms as they're even free running around in their environment. And I want to talk about how we're trying to use these to kind of expand the underlying ideas of what causes aging. My clicker's not working. I'll just stay over here. I want to start by just celebrating what we already know about aging. So the modern molecular biology of aging field really I'd say really got started maybe in the 80s to early 90s. Actually, what really kicked it off in many ways was the discovery that a single gene intervention in C. elegans, knocking down the insulin receptor and some of the other genes in the insulin pathway, could give you a dramatic extension of lifespan. And this was the initial recognition that intervening in these molecular pathways could really have dramatic effects on longevity. And of course, since then, we have the hallmarks of aging, which represent kind of our current best knowledge about things that not only change with age, but that can actually influence the aging process to give us benefits as we get older. So we've made tremendous progress. But let's take a step back and ask how well we really understand aging today. If we've heard about dietary restriction as maybe the best we can do about extending lifespan and healthy lifespan today, we are probably coming up on the 100th anniversary of when we discovered that phenotype, where it was the 20s to 30s where the first observation in rats was made that we could extend lifespan with dietary restriction. I think there was some earlier work way back in maybe some insect models, but that was kind of the real first mammalian system that we found. And just everything we've learned about hallmarks and mechanisms of aging, the best we can do is still not as good as we can do with dietary restriction. So what that tells me is that we still have a long way to go in understanding both the causes of aging, probably some of the interactions between these pathways, and how we intervene to have big effects in aging. So I'm going to borrow the analogy from Matt Kaberlein. You've probably all heard some of this, and make an analogy to our current understanding of aging and where we are in geography. So today we can use something like Google Maps. We can get very deep, everything from the shape of the planets and the continents all the way down to getting directions to something 10 minutes down the road and whether you should go one route or another because there's an accident. We know a lot about how to get around. And of course, that is not the equivalent of where we are with aging science. I'm sure you're all aware. But Matt's analogy here was we're more like the map of Acacius in 500 BC, where we know something about the shape of the continents. We can get around. We know where the rivers are and the cities are. You can use this to navigate. We can use our understanding of aging today to make progress and improve health in model systems. We're starting to get there in humans. But we're missing whole continents where the shape is not there. So we have a long way to go. And what I'm really interested in is if this is what we know, and maybe there's some interactions that we still don't know, and there's a lot of work to do in these areas, a lot of people working on these areas, I'm really interested in expanding the space and really digging in and identifying new areas and things that we don't know. So how are we doing that? My lab uses a comparative biology approach. So oftentimes you don't have to start with human data, but another approach would be to just, for example, take unbiased screen of new small molecules. There's many, many small molecules, millions out there that we could potentially screen. But often we like to start with human data, come up with an idea of a set of genes, a set of molecules, maybe a set of environmental interventions that we think could be involved in aging, but we don't know yet. And then we move into our model system, which is C. elegans. And this allows us to rapidly look at a large number of these interventions and ask whether they can have an impact on the aging process. Once we can then characterize what they're doing molecularly, understand, is this molecule, is this genetic intervention having an effect that might be relevant to humans? If we have confidence in that, then we can move up the chain to preclinical studies in mice, validate that the lifespan extension, the health extension works, that the molecular processes are also conserved, and then ultimately come back and translate these into humans. Why worms in this case? I think there's a couple of distinct advantages here. So I'm going to make a comparison to mice, which is one of our shortest lived mammalian models and one of the major ones in aging research, of course. One problem still with mice, we've heard about dogs and monkeys and the problem with long lifespan. Mice still live 2 to 3 years. That at least is enough that you can fit it in an NIH grant and get money to do this within a couple of years. But C. elegans live about 3 to 4 weeks. So what that gives you is a 40-fold advantage in how quickly you can run these experiments just in the time dimension. A typical mouse study might cost $200,000, something on average. This is to measure lifespan and get some additional phenotype data about health and other things that are going wrong. We can run that same experiment for $20 in C. elegans. That's about a 10,000-fold advantage in cost for running these experiments. So that's the plus side. This, of course, begs the question, if we learn something in C. elegans, how likely is it to translate all the way up into mammals? We don't know the exact answer to that. Even if it were fairly low, this sort of advantage in cost and time savings would be enough to make it worthwhile. And it probably isn't that low. And what we find is even if worms are not going to be the best model for every single disease of aging, it's going to be hard to study skeletal aging in an animal that doesn't have a skeleton, for example. Many of the underlying molecular processes are shared. They have insulin signaling that affects lifespan. They have TOR signaling affects lifespan. They have hypoxia, many of the stress response pathways. Many, if not most of the underlying pathways that are driving aging at this molecular and cellular level are in fact conserved, and we can intervene in this model and in mammalian models and get a lifespan extension. So I think I don't have a percentage of which ones are going to translate, but it looks like it's probably high enough to make this advantage of 40-fold faster and 10,000-fold cheaper give us a big advantage in this initial discovery stage where we can understand new mechanisms and new pathways for aging. I always like to give a little taste of how this is done for those who haven't worked with a worm before. So how do we actually measure lifespan? The old way of doing this was you'd sit at a microscope. This is what the worms look like under a microscope. They're just usually you have 30 or 50 of them on a plate. And you can't just look at a worm and say it's going to live 35 days or a month and a half or whatever it is. You have to go in every day and see if that worm's still alive. So you take a little piece of platinum wire and you smack it on the head and you see if it moves. And then you do that the next day and the next day. And you do that for its 1,000 neighbors that you're trying to study. It's a little tedious. So we and others have built systems that automate this and take this off. So we've basically built a robot. This is a CNC router, so it's used for cutting wood in a computer-controlled manner. You take the wood cutting head off, you put on essentially a microscope, and this runs around and basically drops down this microscope on a plate full of worms inside an environmental control system. And then it takes video of the worms, and that can, using software analysis, we can know when they move. Instead of hitting them on the head with a piece of platinum wire, This system shines bright blue light on them. They don't like that. That has the same effect. They start crawling around. You can see if they're alive. And we can put about 20,000 worms on one of these platforms at the moment. So this lets you do high throughput lifespan analysis and health span analysis. We can get movement data, we can get body size, a bunch of other things. And even more recently, we've just added fluorescence. So now we can add molecular characterization of selected pathways. That's flexible. You can put whatever set of molecular readouts you want on here and get molecular data, lifespan data, and health span data all at once, the same animals over the course of the lifespan. So what this enables is now we can run screens where you can start with hundreds up to maybe millions if we scale this. This system is now scalable. We've taken out that really labor-intensive step of sitting in a microscope and watching the worms, and you can run or you can run a genetic screen. This lets you basically filter out things that are giving you a big extension in lifespan, a positive benefit in aging, and then we can focus the resources at that point. This part's relatively cheap and fast. Then we can focus the bigger bucks on mammalian studies, where those are more expensive, but this helps us focus on the things that are going to work. Just to give you one example, this is a genetic screen we ran in worms where we knocked down a bunch of genes based on human data. These were things that we had correlation with age, but not causative link to age. And one of the top genes to come out was this HAO1 gene. So what this does is it's in the pathway called kynurenine metabolism. You don't need to remember that. Basically, it takes the amino acid tryptophan and turns it into NAD. If we intervene at the point where we knock out HAO, what HAO does is it takes this small molecule 3-hydroxyanthranilic acid, or 3-HA, and degrades it and turns it ultimately into NAD. So we knock down HAO. This gives us an upregulation of 3-HA. And that gives us about a 30% lifespan extension in worms. Either if you knock that gene down, and then we followed it up by just asking, is the 3-HA itself sufficient to give you that extension? It turns out it is. And the worms are transparent. 3-HA is not building up a little bit. It's building up to the point where you can see it like under the microscope when we do this. Might be a little hard to see here, but the worms basically turn red as they get older, and the 3-HA is a nice red color. We've done a bunch of work characterizing this. What it does, it builds up in the intestine. I'm not going to go through all the detail, but it builds up in the intestine, increases intestinal health, probably changes the way the worm interacts with their microbiome to some extent, increases stress resistance, and a bunch of other pathways that we know have some link to aging. So it looks like there's mechanisms for this molecule that are linked to aging in the same sense that we see aging link in mice and humans. Of course, the question is, does it work in a mammal? We've done that. So you can knock out that same gene in the mammal. It's a conserved pathway. Or you can feed them diet. These mice were already old when we started the diet, and we still got a big lifespan effect. So we've now got preclinical data that says we can mimic what we did on the worm in the mouse. We're still working on some of that mechanistic characterization here. It looks like many of the same pathways are being activated. And this is as far as we've gotten. We haven't gotten to the humans yet for this guy. So that was an example. Does it work in humans? So I want to take just a quick step back and talk about the process here. So we are really in the early stages of discovery. My work is at the very foundation of the progress from basic science up to clinical medicine. We start with discovery. That's the screen, mechanism, figuring out how it works. This is really what I'd call the basic science in our simplified view. We've now gone to preclinical validation, and ultimately we want to get to clinical medicine. And this, I would say, is the product or the drug development phase. So here, we've done the C. elegans work. We now have preclinical validation, and the question, of course, does it work in humans? The point I really want to make here is taking a step back to where we started. We have a lot of work going on toward the kind of mid to top of the pyramid now that we have hallmarks of aging. There's a lot of work trying to turn these into practical medicines. But to continue our process in the future, we need this broad-based discovery stage at the bottom to kind of keep up its energy, so that as y'all get closer and closer to medicines, that we can kind of fuel the pipeline of drugs that go into your companies and drive this work. So that is where I sit in the industry, and it's kind of a pitch for additional continued basic funding research, even though we now know a lot more about aging. And with that, I'll wrap it up and take questions. Thank you very much. Yeah. Yes. Yeah, certainly the pendulum has swung recently toward translation. That's been the hot topic, and everyone wants to do translational work. And for better or for worse, the funding pendulum has swung in that direction. So ultimately, I think it'll swing back. This is maybe an argument for at least balancing that out. And I had another thought on that. But where does it fit? So actually, C. elegans, with the regulation, C. elegans actually counts as an alternative model to animals. Even though it's an animal, it's not a cute and fluffy animal. So maybe they don't care as much in the same sense. So this actually has an exception to that, and hopefully we will be able to kind of get through this process as we figure out exactly how we're going to study aging in the future, and C. elegans can be a part of that, I hope. So. Thank you.
Thank you very much.